In Lab 6, we learn about the process of cell division. So for this video, I'm not going to go through all of the steps of mitosis and meiosis because your book has a great outline. The videos that I give you talk about what happens in each of the steps, and you guys are going to be drawing all of these steps. But I do want to do a couple things for you. The first thing I want to do is to outline what happens in mitosis and meiosis as a general process, do a little bit of comparison, and then give you a few tips on how to actually work through this lab. So there are two types of cell division. We have mitosis and we have meiosis. And they each serve different purposes. So overall, the purpose of both of these is cell division. We're going to make new cells. But the goals of these processes are slightly different. So the goal of mitosis is basically repair, replace, and grow. So cell division happens through the process of meiosis to repair damage, to replace worn out cells, and for the organism itself to grow. Mitosis is essentially asexual reproduction. Now, single-celled organisms such as bacteria, single-celled prokaryotes, reproduce by a process called binary fission, which is sort of similar to mitosis. The steps are different, but they both sort of fall into this idea of asexual reproduction. The goal of meiosis, though, is to produce gametes. Those are the sperm or the egg cells, or whichever the respective cell types are in other organisms for sexual reproduction. So this is reproduction in which you have two organisms contributing genetic material. So our goal of mitosis is repair, replacement, and growth. And so this happens in our somatic cells. So these are all the cells that are not our sex cells. So all the cells that are not sperm and eggs. Meiosis happens in our gametes, which are our sperm and eggs. So let's talk about some of the differences in this process. I'm going to relabel mitosis and meiosis since you can't see it. So meiosis starts with a diploid cell, that it is a cell that has two copies of each type of chromosome. Now meiosis also starts with a diploid cell, cells that have two copies of each type of their chromosomes. The end of meiosis, we are going to have diploid cells. So in mitosis, we maintain chromosome number. At the end of meiosis, though, we produce haploid cells. We have one copy of each type of chromosome. So at the end of meiosis, we have reduced chromosome number. And this is because when you join a sperm and an egg, when you join their genetic material, you double it. And so you have to start out with half of what you need in the beginning. In mitosis, we start with one cell and we produce two daughter cells. And in meiosis, we start with one cell and we produce four daughter cells. Now, the reason for this is that reduction in chromosomes. We need a second cell division in order to reduce our chromosome number to haploid. And then one of the most important differences in mitosis and meiosis is that mitosis produces genetically identical cells. All the chromosomes should be exactly the same as they were in the parents. But in meiosis, we produce genetically unique cells. And this is because in sexual reproduction, we introduce genetic variation into a population. So one of the key differences in mitosis and meiosis is this production of genetic variation. So let's summarize those key differences. Well, not summarize. We're going to continue to summarize here. Mitosis has only one cell division, whereas meiosis has two cell divisions. In mitosis, we separate sister chromatids. I'll review those in just a minute. But in meiosis, we separate homologous chromosomes in meiosis 1, and then we separate sister chromatids in meiosis 2. We also have two processes in meiosis that lead to that genetic variation. We have crossing over, and we have random assortment. So I discussed both of those in our lecture video, but if you have questions on how those work, please let me know. So as you're working through this lab, we're going to ask you to draw pictures at various steps in this process. And we're also going to ask you what's happening in some of these steps, what key features are happening there. And then in many of the steps, we also ask you to count the number of chromosomes. So I would like to review the types of chromosomes that you're going to see and how we count them. So there are essentially two different versions of chromosomes. We have homologs or homologous chromosomes, and we have sister chromatids. So let's start with homologous chromosomes. So homologous chromosomes are chromosomes that come one from mom and one from dad. Or I should actually say maternal and paternal. Paternal means it comes from dad. So homologous chromosomes have the same physical characteristics So their arms are the same length, their centromeres are in the same position. If you stain them for a karyotype, they produce the same banding pattern. They're the same length. They have the same gene order. But they may differ in the alleles for those genes. Now, they might be the same for certain alleles, they might be different for others. There are all sorts of combinations that could happen here. So homologous chromosomes have the same physical characteristics, the same gene order. They come from the fertilized zygote that is from your parents. So mother's egg combines with father's sperm. They combine their genetic material. Each parent contributes 1/2 of that homologous pair. Sister chromatids, however, are produced from those homologous chromosomes in DNA replication. So DNA replication happens we make an exact copy of those maternal or paternal chromosomes. And for the time being, in the S phase of our cell cycle, those chromosomes remain attached at the centromere. Now, this is so they can stay together, so they can stay organized for cell division. So these sister chromatids are identical to either the maternal or paternal chromosome that they were copied from. So essentially, they should be 100% identical copies. So mom's chromosome, chromatid number 1, should be exactly the same as chromatid number 2. Now, they may differ only due to errors in replication. Okay, so you might have a few sequence changes, but for all intents and purposes, we are 100% identical. So if you have allele A here, you have big A allele there. If dad had little a allele, he has little a allele there. So if we are thinking about gene A, we might have different alleles in the homologs, but our sister chromatids are going to be 100% identical. So how do we count this? So let's count some chromosomes. You count chromosomes by counting centromeres. So here we have 1, 2. Our chromosome count is 2. We have 2 chromosomes. Here at our sister chromatid side, we still have only 2 chromosomes. On this side, we have 2 chromosomes. On this side, we have 2 chromosomes, but we have 4 chromatids. So you have to make sure that you have this vocabulary straight in your mind. We have 2 chromosomes or 4 chromatids. So let's draw just a sample cell. I'm going to draw some homologous chromosomes. We're going to say the red and the pink are homologous, and then we're going to say the blue and the black are homologous. I'm just trying to get you some color so you can see sort of what's happening. So if this is our cell that we're starting with, we have 1 homologous pair, 2 homologous pairs. We have 2 copies of each pair. That means that this cell is diploid. So our chromosome count is going to be 2n. We have 2 copies of each type for a total of 1, 2, 3, 4 chromosomes. Now let's say that we go through meiosis. We're going to end up with 4 cells, each of which have 1 copy of each type. So each of these cells have 1 copy, 1, 2. They are haploid, where their chromosome count is n equals 2. So let's look at some sister chromatids. Actually, let me come back here. So in this process here, we went through meiosis, but here we're going to go through replication. I actually jumped ahead. I meant to do replication first. So we're going to replicate these chromosomes and produce our sister chromatids. So what is our chromosome count now? Well, we are still diploid. We still have 2 copies of each type for reddish colored chromosomes and our blackish colored chromosomes. So we are still diploid. We still have 2 copies of each type, and we're going to count our centromeres. 1, 2, 3, 4. So we are still 2n equals 4, even though we have done replication. We have increased chromatids, but we have not increased our chromosome number. So let's look at a couple examples. Humans have 2n equals 46 chromosomes. That's our diploid number. That means we have 2 copies of each type of chromosome for a total of 46. So our haploid, our sperm or our egg cells, is going to be n equals 23. That means essentially we have 23 types of chromosomes. So this is humans. Let's look at some other organisms. Let's look at chimpanzees. Chimpanzees are 2n equals 48. That means their haploid number is 24. So they have 2 types of chromosomes. 2 copies of each type. They have 24 different types. Let's look at some animals. Horses are 2n equals 64. That's a lot of chromosomes. So their haploid number is n equals 32. So let's say that we have a horse, and that horse mates, and we have an egg where n equals 32 and a sperm where n equals 32. After fertilization, we restore our diploid cellular makeup, and we get back 2n equals 64. Let's go backwards. Let's say we have a fruit fly who has produced eggs, and another fruit fly who's produced sperm, and we let them mate, and they combine their gametes that are haploid with a chromosome count of 4. That fertilized embryo is going to be diploid, where 2n equals 8. So we can go both ways between this diploid and haploid. So finally, let's get to the lab. You are going to be making chromosomes out of these little beads, and you are going to have, you guys are doing yours a little bit differently than colors just to keep things straight. You're going to make 2 types of chromosomes. Verify in your lab manual the number of beads that you're supposed to make. Sorry, this one should be red. So we have 2 types of chromosomes here. We have large chromosomes and we have small chromosomes. These are your homologous chromosomes. All right. Early on in your lab, you are going to do DNA replication. And when you do that, you're going to produce sister chromatids for each type of cell. Sorry, each type of chromosome. Okay, so there's our large one. These are still homologous chromosomes, but now we have made sister chromatids. I'm just going to call them sisters there. So let's replicate our other set of chromosomes. And these are magnets, so these centromeres are going to stick together. Again, these 2 are homologous. These 2 are sister chromatids. So you are going to be moving around these sister chromatids, following the instructions in the simulation. There's a video on the simulation. You can also use your book to help guide you for where you're supposed to be placing these. If you're doing this online, we've actually done the simulation for you. You just have to draw it. But the important thing there is that you keep track of what is moving and how it's moving. So you have to pay attention to how things have moved around. I do want to point out number 13 in your lab manual is asking about random assortment. So often, students will be drawing their chromosomes along an equator. Don't move your equator. Do not flip it this way on this question. I'm not going to tell you what you have to do in number 13, but I'm giving you a hint here. Keep your equator in the same place. Now, in order to keep your equator in the same place and do a new assortment of chromosomes, you have to understand what's happening in random assortment. So if you have questions on this, please let me know. You can check your book. I talk about it in lecture. Your book also talks about it. And your book gives you examples of different random assortments. So my other suggestion is just keep your equator the same throughout your entire lab illustration so that you can see how those chromosomes are moving. Two more last little pieces of advice. Draw your chromosomes carefully, label what it asks you to label, pay attention to your numbers, but don't spend a million years making this look absolutely perfect. I am not grading your artistic ability. I need your drawings good enough that I can tell what's happening, but you don't have to spend a million years making them perfect. Make sure you read your instructions carefully so that you label everything that we're asking you to label, answer all the questions that we're asking you to answer.